Okay, so let's get started slowly, and then people can watch the recording if they miss some pieces of it. Um, so as I said, our meeting is recorded. We typically post VOOS on YouTube at the end of the day or the next day. If anything happened during the meeting today that make it so that you prefer that we make some edits, please reach out at the end of the meeting so that we can adjust. Um, okay, so the meeting agenda today is to first, I thought it would be helpful to share an update on the physiological review submission and have potentially answer any questions or discuss the strategy to move forward. And then in the second part of the meeting, I'm hoping that we finalize the pilot experimental design. Because we are getting really close to running this experiments and we need to finalize some blocks. So that's last week we agreed to have an unstructured discussion today. So any other things we should cover during the meeting today? Okay. So let's go on the physiological review. So what we've done in the last few weeks is physiological review is a journal that is an invite only. So their process is typically to um, select specific topics of really broad interest in the community, and then find leaders in the field to write they call it a definitive review on the topic, and then and work with these person to publish it. So our process has been different than their typical process for writing reviews, and so what we did in the last few weeks is submit. So we reach out to the board, um, um, explain our review piece and how it was written, and then submitted an applications to their board so that they could review and decide whether they wanted to move forward. Um, a particular challenge was that they wanted to have senior authors are as co-corresponding authors, because that's not my I'm not a senior according to their criteria. And so as a group we agreed in the last month to had Henry Kennedy to as a co-corresponding author, and then to eventually discuss with them whether it will make sense to add additional co-corresponding authors, potentially across the different sections, um, because it will be helpful to go through the review. And so in the last few weeks, we submitted the first proposal to the board. They review it, and Henry and myself last week received a formal invitation from Physiological Review to submit our piece. So that means that they are interested in what we wrote or the proposed topic, and the coverage of it. So that's a good news. It means that the journal will have a high likelihood of taking a piece, provided we go through review. Um, so I exchanged a few emails with Henry Kennedy last week. Um, remember that we talked about potentially adding more corresponding authors. So our strategy will be to potentially accept their invitations and discuss adding potential additional corresponding authors. Henry is currently traveling, so I haven't been able to go in depth into this topic, but that's currently where we are. The other aspects is the manuscript length. So in their invitation letter, they said that they typically publish 40 pages pieces. And so we are likely twice bigger. Uh, and so what I think we need to do is probably measure exactly the length according to and they provided some guidance on this. Measure the length according to their formatting criteria, and then in our emails ask them whether they have some flexibility, because they are asking for writing a very definitive review. So that means something that has a really good coverage of the predictive processing literature, and that literature is so large. But I think this calls for what we actually wrote. So I would like to try to minimize the number of edits that we need to make. But we need to get their buy-in on that. But so that's where we are. Any questions or comments on this? I don't hear any concern. Okay. So I'm going to move forward with discussing with Henry Kennedy, and then once this occur, I think we'll probably write a message to everyone to keep everyone up to date about what's going to happen. I'm hoping we can submit the piece as close as is as possible, but it's possible that we need to do a round of edits. Particularly, we might need to sort of consider missing publication that came out since, because the reviewers will likely call it out. Okay, before we move on to the next topic, have any questions? Okay, so the second discussion we wanted to have is so last we've been talking about including so that's our current stimulus design for the pilot. So remember that we are implanted mice already to go through the NeuroPixel recording platform, and Boost will start. I mean, in principle, the first recording will start in I think in a month. But the mice have to be trained on the visual stimuli starting 1 or 2 weeks from now. The first mouse I went through the pipeline had some visual area targeting issues, so we decided to not use this animal. And so it's likely that the first mouse on the cluster, the training cluster without neural pixel recording, will start in 2 weeks. And so our stimulus, the big part of it is ready, but the things that we need to finalize is the final block that we have been talking the last few weeks. So this is Zebra, Trippy, and potentially Gabor patches. So last week, Dimitri presented on Zebra, and our proposal is to have 5 minutes of Zebra, 5 minutes of Trippy, and some Gabor patches to compare for the receptor field mapping code to make sure that boosts are properly working. So what we did in the last week is so we've collaborated with the Zebra team, actually have now Google Colab to recreate a Zebra movie. It's in the discussion forum. So we should be able to generate that movie with the rig geometry. So it's positioning the screen 15 centimeter from the eye, as opposed to 10. So in the Zebra paper, there were 2 screens at 10 centimeter. So we adjusted there. So you can follow this in the discussion thread. On Trippy, the challenge there is the Micron's team use 15 seconds long trials that are appended to one another. And that accounts for 10 short trials that are presented twice in the Micron dataset across multiple sessions. So that would create the same stimulus that was presented to the mouse with the functional connectomic data. And so I was trying to regenerate Boost movies. The data is the seeds to generate the Trippy movie is in their database, and so I was just able to access this yesterday. So we're working with Alex to regenerate Boost. But so my current plan is to implement these design, But I wanted to know today if there was any feedback or opinion on how we should adjust this design. Any particular concern or no feedback? It sounds like you're all on board. I think Alex was Alex, are you here?
Yes, I am.
Yeah, are you on board with that, Pan, or do you have any concern?
Sounds all good.
Okay, okay. So I mean, based on if you don't have any feedback, I'm going to implement this. So uh, with Alex helps and anyone else. Okay. I mean, I was expecting some discussion, but if everybody's on board, we don't need to dive deeper into this. One other thing that is happening today that I thought it would be helpful to share is Rafael Bugaz, Claudia Klopath, Zenke Friedemann, and Jörg Keller are actually visiting the Allen Institute. And so this afternoon, we're going to share with them, we're going to show them some of the setup to record the dendritic activity as part of this project and potentially collect any feedback they may have on this experiment. So I'm just leveraging the LEC conference opportunity to disseminate our effort a little further. So you might hear from them in the next few days. Doesn't seem like there's much feedback today. So next week, we were planning to have a SLAP2 focus session. So Jesse is preparing some analysis of the SLAP2 data that we collected. I know Lucas agreed to it as well as Sarah. Um, are you going to, can you confirm these? I don't know if Sarah, you can hear us? Maybe not.
You put some questions in chat.
Oh, SST up to tagging. So Sarah, the initial design with NeuroPixel recording was to do SST tagging. So because we have the ability to identify SST cells with NeuroPixels, and so we were planning to do that. It's in the document that we wrote. So this is not for only the pilot. It will be also for the final data set. But it takes 10 minutes to run. That's right. And sorry, you'll be able to present next week on SLAP 2, or how much time would you need? Okay, well, she said 15 minutes. If there are no more comments today, we can adjourn. Okay, so what was the short meeting today? We'll regroup next week for discussing the SLAP 2 data analysis. in depth with multiple presenters. Thanks, everyone.
Good seeing you, Jerome.
Thank you. Bye.